And I never thought there'd be, uh, I never thought there would be employment in it. It's a strange thing, you know. I thought for sure that I'd end up stamping dock approval permits for the Ministry of the Environment or something. But I was so excited when I was able to find out that I could find employment looking at fungi and studying fungi. And it's been it's been over 20 years now since I graduated. And it's been a really amazing ride and an amazing run. And I see so many amazing things in the world of fungi, and so I'm happy to share them with you tonight. I put this presentation together. You know, there's an awful lot of resources now online about mold. And I've made sure I spelled it here for the American audience. In Canada, we would spell it M-O-U-L-D. It's a common British spelling, but I spelled it M-O-L-D for this presentation. I think most of our participants are American, but either way is good. There's no judgment. And I really want to look at tonight what are the organisms beyond just the molds. You know, I want to look at a fleshy ascomycetes, basidiomycetes, more than just the mold that grows in buildings, because there's so much more that grows in our indoor environments. So, you know, so to compare and contrast, so this first image in my title screen shows us an awful lot of mold colonies growing on a ceiling, on drywall, on wood. But this would be a much different image. This would be an image of basidiomycete growing on timbers, on structural timbers. So a very, very different organism. This is not mold growth. You know, this is indoor fungal growth, but certainly not mold growth. So what is it, and how do we wrap our minds around how these organisms interact with our built environments? Who cares about indoor fungi? Well, it is important. You know, indoor mold growth certainly can impact the health of human occupants. We know that people with asthma are bothered by indoor mold growth, and even for people not suffering from asthma or afflicted otherwise, will have a potential respiratory irritation, a runny nose, red eyes, you know, other issues associated with respiratory irritation. It's not a very well studied area of public health. We certainly have sort of come to the conclusion as a society that we should avoid and eliminate indoor mold growth, but it's very difficult to understand the specific impacts of a lot of fungal indoor growth on human health. There are opportunistic human pathogens that we have to consider. So when we think of bird droppings or bat droppings, guano, you know, we have things like histoplasmosis to think about. We have a fungi like Aspergillus fumigatus that can grow on a number of substrates in the indoor environment, which can cause opportunistic lung infection. So these are all things that can affect even normally healthy people, but most especially people who are immunocompromised. Wood rotting fungi. This is a really fascinating one to me, and that's the one slide we saw there. Wood rotting fungi can cause structural damage to buildings. You know, we build a structure, we engineer it, and we have a load-bearing capacity that we assign to structural wood in the building, but a fungus comes along and then strips that wood of its load-bearing capacity. So it impacts the engineering specifications of that building, and that can lead to really traumatic and significant failures of the built environment. So the types of fungi found indoors, not surprisingly, is very similar to the types of fungi we find outdoors. So we certainly find basidiomycetes, we find ascomycetes, we find zygomycetes, anamorphic fungi, which are, so molds are asexual fungi imperfecti. There's a number of different ways to refer to molds. But we typically in the indoor environment, we really do find anamorphic fungi more commonly than the teleomorphic fungi. So the asexual state is more commonly present, from my experience anyways, than the sexual state of fungi. And we certainly find myxomycetes are not properly fungi, but we do find myxomycetes and slime molds in the indoor environment as well. So mold and mushrooms. Sorry, some of these things here may seem simple to some, but maybe very new to others. So I'm just making sure we cover all our bases for everyone who's participating tonight. So fungi reproduce either sexually So they might produce a mushroom or a fruiting body by way of sexual reproduction or asexually. And so molds represent a form of asexual reproduction for fungi. Sexual reproduction is typically more, takes more time and energy. It requires more resources. It's more energetically expensive. So generally fungi, insofar as they may opt in a scenario to produce their sexual structures, they want to know that there's a more reliable resource base available. If there's just a short-term availability of core components like water or substrate, insofar as the organism is influenced to make a decision or to opt to produce either a sexual or asexual state, you'll tend to find the asexual state when there's not a lot of resources available, or if it's a highly competitive environment. Asexual reproduction is faster. It has lower energy requirements because mold can grow and sporulate relatively fast. We can get, from the time of an available scenario event, so having the right amount of water in an environment and a substrate to grow on, a mold can often start producing spores in less than 48 hours. It would be very unusual to get in an indoor environment mushroom production in under 48 hours. That typically requires a longer time span to achieve. So our big two in fungi, our big two divisions, ascomycetes and basidiomycetes. So ascomycetes, when they reproduce sexually, they make their spores in a sac, an ascus. So an ascus, an ascomycete. It's spores in a sac. We're going to see some pictures of that in a bit. It'll make a lot more sense. And basidiomycetes, so when they reproduce sexually, they produce their spores individually on basidium, on basidiae, and we all know, those of us, most people here, I'm sure, are collectors. They're in the forest. They're forayers. We all know the button mushrooms, the porcinis, the oysters, the chanterelles, the portobellos. Either if you're buying them or you're collecting them in the field, it's really basidiomycetes that we generally, as weekend collectors and forayers, tend to collect more commonly. Indoor basidiomycetes. How we encounter indoor basidiomycetes most commonly, from my experiences, as wood rot fungi. And the affected materials are usually in our built environment, in our homes, in our offices, in our churches, in our courthouses. Our wood construction components, the framing, the floor joists, the beams, the subfloor oriented strand board, plywood. This tends to be where they find their home. They generally require a stable and sizable supply of water to sustain growth. So a single incidence of a weekend, a short-term flood that lasted in a basement for 24 or 48 hours may be less likely to cause the growth of basidiomycetes indoors, whereas if there was a 2-week flood or a 1-month flood or 2 months of increased humidity, high humidity in a space, then we may see that type of growth more commonly in that scenario. They do tend to be large in size. They're very successful. Indoor basidiomycetes wood rot, when they do set up shop inside the built environment, when they do take root, they're very successful often in colonizing a very wide and large area. Dry rot, which we're going to look at in a few moments more specifically, dry rot digests cellulose, and it leaves wood with a greatly reduced structural strength. So the big problem really, when we think about indoor wood rots and rot from a basidiomycete, tends to be dry rot, because when you take the cellulose out of a material, that tends to be quite impactful on the material's load-bearing capacity, which is an important engineering characteristic. This is a picture of a mushroom and a potted plant. Many people have seen this. I think that's probably a leucocoprinus. I didn't get this picture myself. I think leucocoprinus is the common mushroom that a lot of us find growing on indoor plants. So we do find gilled mushrooms. That's not a wood rot fungus per se. That's a gilled mushroom growing in the indoor environment in a plant. It's just all opportunistic. Things that grow outdoors will grow indoors if we give them the right conditions for growth. All right, so I want to look at one of the items I want to look at, and this is going to be a gilled fungus here. It's not a dry rot or white rot or brown rot. This is coprinus growing beneath a subfloor that I encountered in an environment. And again, I'm looking at the robustness and the variety of fungi we find growing in the indoor environment. So I was working on a job where there was air samples for fungi related to a pipe leak, a water pipe leak in a kitchen in an elderly home care facility. And there was a lot of counts in this. There was, of course, there were things like aspergillus and penicillium. There was lots of mold spores, but there was an awful lot of coprinus-like basidiospores. And so what was interesting about that to me is that sort of indicated that we have something more than just our common suite of molds. We have an entrenched basidiomycete growing somewhere. And generally, if the basidiospores are there, well, you have to have the fruiting bodies. The mushrooms have to be there if we're seeing the mushroom spores. So if we have coprinus spores, we're going to find those mushroom fruiting bodies somewhere. In that area, the kitchen area that was impacted, there was visible mold growth, but there was no mushroom fruiting bodies. So in scenarios like this, in my line of work, it's a little bit of a Sherlock Holmes type of a scenario. You got to put on your cap and you have to look and see, well, where is this? Where are these basidiospores coming in from? Where are these coprinus spores coming from? And of course, as often is the case, a trip to the crawl space, we were able to find this, beautiful fruiting of coprinus. Now we're looking at in this picture here of a fruiting of coprinus basidiomycetes fruiting bodies in a space. But if you look at, and I want you to appreciate in this shot, the entire darkened space of that subfloor. So I think it was an OSB subfloor, so an oriented strand subfloor. That's the type of wood that's being supported in that image by the timbers that you can see, by the joists. It was entirely covered with the deliquesced spores of coprinus. So all that darkness that you see along there is just a mass of coprinus spores. So we're not seeing a one-time incidence of a fruiting of coprinus. What we're seeing here is a repeated pattern. When we're seeing this image, this has happened so many times previously that the entire subfloor there is covered with coprinus spores from deliquesced coprinus growth in that space. So it's been going on for an awful long time. It's just been undetected. Nobody noticed it. It happened all beneath the floorboards. But in looking at the air samples in the space above, we were able to capture that and then know to look for something more in the area around what was our sort of prima facie, our initially impacted area. Okay, so wood rot. Wood rot's really the big one. Besides, finding those guild mushrooms is more of a rarity. Wood rot's this thing we're going to find more often insofar as basidiomycetes occur. Wood is made up of two key components. So I want to look at wood for a second. So when we think of wood, the two major components of wood are cellulose. So those are the long fibers that impart strength to wood. And then there's lignin. So lignin is a glue. It's the binding matrix in wood that keeps all those long cellulose fibers together. Those are the two major components of wood. And that's important in how we look at the rotting of wood, because as it turns out, fungi tend to be fairly specific in how they rot wood. So a white rot fungus, when we go out into, and then we see this in the forest. This isn't just in our buildings. We're going to see this when we go out to collect mushrooms this weekend. We're going to see white rot fungi. We're going to see brown rot fungi. So a white rot fungus, they consume the lignin in the wood, and they leave the cellulose behind. So the wood actually keeps a lot of its strength. A white rot impacted wood actually is fairly strong still, but we can still tear it apart. It's not as strong as it was with the lignin, but it's relatively strong. And the wood, when we see it, often has a bleached appearance. I often see this in the forest. There's species of Nectria. So when we go out on the weekend to collect fungi, there's species of Nectria. I think they call them white face rots. And we often see that where a branch has been removed from a tree, either by nature or by man. And we'll see that it's white rotted and it has that appearance. It's bleached out. Here's a picture here of white rot affected wood. So you can see this wood here. Looking at its texture, looking at its composition, it's not as strong as regular wood. You can actually use a knife or a thumb to pry it apart. It tends to break apart in long segments because the cellulose fibers are still intact, not as strong without the lignin, but still fairly strong. Okay, so then there's brown rot. All right, so when we think about brown rot, there's fungi that go into wood and they consume all the cellulose instead and leave the lignin behind, right? So again, very specialized, fungi. I love fungi. You know, there's over 1 million species. We hardly know any of them, and they're very, very specialized, amazing organisms. And so the brown rots go in, take the cellulose, leave the lignin. The brown rotted wood breaks easily into cubes. And so often when you see this in the forest, you see a very cubic appearance, a dark brown cubic appearance of the wood. And it doesn't really have much strength. Oftentimes you can pick it up and crumble it in your fingers. It's very much like styrofoam. You certainly wouldn't want to stand on it. You wouldn't want to cross a stream on a log that had been rotted by a brown rot. So you'd be in that water, absolutely. It would have very, very little load bearing capacity. And then of course, we're going to see sometimes mushroom growth associated with the wood rot. And it's not, of course, mold. Everyone very much thinks typically of indoor fungal growth as mold growth, but this is fungal growth, but not with mold, not necessarily with the same spores, of course, that we find with molds. All right, so here's a picture, and this is a picture from an indoor environment, but you're going to see this in the field as well. When you go meet, I'm a big fan of myxomycetes. I'm always rolling logs and looking at logs for myxomycetes and ascomycetes as well. I'm a big fan of the ascomycetes in the woods. And so you're going to see lots of logs in the forest that look like this. Hopefully you don't see any timbers in your home that look like this, but you can, it does happen. But this is ideally something you'll see in the forest. So this is a brown rot impacted timber, and you see that cubic structure. You see how it's breaking apart and fracturing in a very cubic fashion. So the cellulose is being eaten by a fungus and only the lignin remains. So what we're looking at there is just the remaining lignin. All right, so I'm going to talk about a case that I bumped into that was really fascinating and interesting. And it came about because we were finding on air samples basidiospores, so mushroom spores from a basidiomycete in February. Now, I know a lot of you are from all over the United States of America, but I want to mostly talk now to the people from the North, the Canadians, the people from the Northeast. In February, there's snow on the ground. We're not finding mushrooms growing outdoors in February. Typically, indoor air reflects outdoor air. So we shouldn't really be finding a lot of basidiospores in January, February, December in Canada, in the northern United States. It's winter, and so it's very uncommon. So it was in a century-old church that we were finding this, and it was reported as a roof leak. So what led to the investigation of indoor mold growth was a roof leak, and there certainly was visible mold growth on the walls of this church. It's an old church, so this is I think it was built in sometime between 1850, 1875. And of course, that's a plaster interior. And so the plaster that had been impacted by mold had been removed. Air samples have been collected in the affected area. So when they're doing a mold remediation or a mold abatement on the building, they build a structure called containment. We're going to look at that in a second, around the affected area. And they had high counts. So this is in February, high counts of basidiospores, about 3,000 per cubic meter. Now, I didn't get to look at the initial samples, and I would fault the analyst probably who looked at that initially for not pointing out that two things. First of all, that's unusual to find at that time of year, and second of all, that they're all the same type of basidiospore. Usually in the summer when we see a lot of basidiospores in the air, there's a wide mix of spores. Because the indoor air is drawing from the outdoor air, and there's an awful lot of basidiomycetes out there producing spores. So we should see normally a nice wide range of fungal spores, of basidiospores in the sample. This was all very much monotypical. There was one type of basidiospore showing up. And of course, it's February. And so when we look at the built environment as building mycologists, as building engineers, when we look at the indoor air, what we're always comparing it to typically is the outdoor air. The idea is that indoor air should look like outdoor air. The air in our homes should look like the air outside of homes. We open our windows up, we draw air into our homes. It probably should have a lower total volume of material, but it should qualitatively look the same. Even if it's quantitatively lower, there should be qualitative overlap. But why are we finding all these basidiospores in February? That's a bit of a problem. So this is inside of the church, that area there that you see with the poly plastic containment built. They build a containment to control the air around the affected area. So that's the affected area behind that plastic sheeting that we're looking at. It's inside a church. So this, now we're looking right now at inside that containment. So here I've got a picture here from inside the containment. We can see they've removed the plaster. So we can see the wood lath behind the plaster. And if you can see there, and we just looked at it previously, what does wood rotted by a brown rot look like, right? It's eating the cellulose out of it, and it's leaving this very cubic sort of appearance behind. That's a closer up shot there. So we can see right here, of course, we can see that the structural wood of this church is being impacted by a brown rot fungus, right? I don't need to see spores when I look at this. I can tell just by looking at the wood qualitatively that it has been impacted by fungal growth. It has been degraded by a brown rot fungus. It doesn't matter if I see spores or mycelia or hyphal fragments. All of these other considerations are secondary. I can tell by the material itself that's been impacted. And then this is from above the impacted area. And I always think of, it's a funny thing. There's probably a lot of you that enjoy honey mushrooms. So Armillaria mellia. And it's interesting, when we think of honey mushrooms, we who collect, I'm actually not that big of an eater. I will opportunistically collect for food. But we think of, Armillaria mellia as a honey mushroom. It's interesting, if you ever work with people in the forestry industry, they call it the black shoestring rot fungus. That's much less flattering, isn't it? When you eat the fungus, it's a honey mushroom. Oh, it's honey, that's delicious. But when it's killing trees, when you're worried about the timber value of a woodlot, it's the black shoestring rot fungus. And then that, of course, comes from the presence of rhizomorphs. So these fungal structures that work through the substrate to gain nutrients, to gain water. They're working to draw back resources, nutrients to wherever the central structure, central command of the fungus is growing on that particular substrate. So we can see here in that yellow circle that pattern. It looks like root-like structures, fungal rhizomorphs, those cord-like mycelial structures growing across the brick surface. So again, that's proof positive of fungal growth. Doesn't matter about spores. I don't need to see mold. I know there's fungal growth there based on the presence of those structures. All right, so in the containment status, when we look inside there's no visible mushroom in that containment. So we do, though, have an issue of elevated basidiospores to consider. So when we collect an air sample in that containment, we're getting basidiospores. I don't see a mushroom anywhere in that containment. And we have brown rotted wood present. We have fungal rhizomorphs present. So we know that in terms of saying that particular area is no longer impacted by fungus, that the remediation contractors have gone in and they've done their job and they've removed the impacted materials, this is a fail, okay? Because we have wood that's impacted, we have rhizomorphs, there's fungal material still present. But the real question is, to me, since in the air samples we're getting basidiospores, where's the mushroom? All right. So as it often turns out to be the case in these scenarios, you talk, again, you put on your Sherlock Holmes hat and you talk to the workers there. And one of the workers there mentioned to me, he thought he saw something that looked like a mushroom underneath the church. Now, this church didn't have a proper basement. It wasn't like an 8-foot passage that was well lit. It had a crawl space that was anywhere from 2 to 4 feet probably in height. And so, but I'm a committed investigator. So I went down there with my flashlight and with my knife. You never know what you're going to bump into down there. I'm always afraid of raccoons or something down there. You never know what you're going to bump into. But down I went, and of course, as soon as I went underneath, I started finding impacted timbers underneath the floor of the church. So here we're looking at joists underneath the church. So they're very much impacted by brown rot. And that knife there, because it's like styrofoam, sliding my buck knife there into that joist, that structural supporting timber underneath the floor of the church. It was like sliding it into styrofoam. It really doesn't have a lot of structural strength left. It's been very much impacted by the fungal growth, and its load-bearing capacity has absolutely been compromised. So while I was down there, it's an interesting thing. I turned around at one point and there was this wall of smoke or fog approaching me. I was underneath a church. I thought, oh my goodness, what is that? It's a very scary. All I've got is a knife and a light and a wall of smoke approaching me. It turned out though it wasn't the devil himself or Satan coming for me. It was just there was a pressure release valve. So this old church had a boiler system and radiant heaters in the church. So it's a water-based heating system. So it has pressure release valves underneath the floors. And this is probably a terrible idea. It really is. Because if you think about it, you're releasing all this steam and all this moisture onto wood. What a terrible, terrible idea. So let's have a look. So this is the wood now with all the steam being released underneath the floor of the church. And you can see the water just building up there. My goodness, this is absolutely the last thing we want to see in the built environment is the thoughtless or indifferent addition of moisture to cellulose containing materials, to wood, to drywall, to plaster. This is not what we want at all. This is a fail right there. And so I looked a little further, and this was interesting to me. There was a foundational stone in the one corner of the church, and that's where I found the fruiting body of a fungus, of a basidiomycete called Serpula lacrymans. Now, Serpula lacrymans is largely considered in Europe, especially Northern Europe, to be the dry rot fungus. This is considered the most problematic of all the dry rot, of all the brown rot fungi you can get. And here you can see, just to give you some perspective, that would be the fruiting body was about 3 feet in diameter. So it's a resupinate, so it's a polypore. It's a resupinate polypore, and it's growing on stone. That's interesting too, isn't it? Because you think, oh, it would grow on the wood, right? If it's using cellulose, to feed itself, it would fruit on the wood, wouldn't it? But it didn't. It fruited on the stone. So it's a 3-foot wide diameter fruiting body on a stone, on a foundational stone of a church. That's an interesting thing. That's the leading edge of it there. Serpula, I always say, I love, the names of fungi. Serpula, so Serpula, serpent, and lacrymans is tears. There's probably a lot of exudate associated with the fungus. So if I was thinking about the etymology of the name, it's probably the weeping serpent. It's this very serpentine type of hymenium, the fruiting body, the surface of the fruiting body, and it produces a lot of exudate. So it looks like tears for those of you that see a lot of fungal exudate on basidiomycetes. So Serpula lacrymans, one of the many brown rots that can affect timbers and building. It's sort of the dry rot. It's interesting when people talk about, they just say the dry rot. They're usually referring to this. There's a number of brown rot fungi that can operate in lower than ideal moisture environments, but this is certainly the one, I think, especially in Northern Europe, that claims that title because it can persist through low moisture cycles. The idea of it is that, if you have occasional wetting, sometimes a fungus like this, Serpula lacrymans, can entrench itself, and then during a moist cycle, and then there could be a dry cycle, but it continues either to exist, either by liberating moisture from the cellulose that it digests, or drawing it in from elsewhere. Remember those beautiful rhizomorphs on the brick wall? It can draw in resources from elsewhere to support itself. They're very effective in obtaining water from the environment. And we see that the mushroom growing on wood, we see it growing on stone, we can find it growing on brick, can find it growing on mortar. So why grow on a stone? And that was something that really fascinated me, because I hadn't really contemplated a mushroom, a resupinate basidiomycete, a resupinate polypore growing on stone before I bumped into that Serpula lacrymans. But if you look at it, if you think from an engineering perspective, what's fascinating is as a moisture reservoir. Okay, if we compare timbers in our built environment, if we think of our joists, our beams, our building wood, and we compare it though to mortar, to brick, to stone that we use in our built environment, stone, brick, and mortar do a much better job of holding moisture. It's a very reliable source of moisture. So if you were thinking, where would I build my house? Where would I build my house? I can draw in, I can digest cellulose, and send my rhizomorphs out and bring back, the sugars that I need from digesting cellulose to support my house. But I need a good, ample, reliable supply of water, and I can often best get that on stone or brick or mortar. So I'm going to build my house. I'm going to build my fruiting body on stone, on brick, on mortar. So it's an interesting thing, because usually when we think about mushrooms growing in the forest, the mushroom is growing on its substrate. It's digesting the substrate that it's growing on. But in this particular case, the fungus is actually producing its fruiting body not on its dinner, but on its drink, right? So it's building it from on the place where it's getting its moisture from. That's a really unique and interesting and fascinating thing about Serbia. Okay, this is a picture I showed. This is my second picture in the slide. I always love this picture. Now, I didn't get into this environment. A colleague of mine sent me this picture, and you can see here just massive mycelial growth, draping down from structural timbers. This was actually in the basement, as I understand it from my colleague. This was in the basement of a mental health care facility here in Canada. And there's chains there. There's these things running on. It looks like something from a science fiction movie. Really amazing growth. I didn't get to look at it directly, and apparently, nobody in the remediation process ever bothered to try to identify it, which is fine. That happens sometimes. But looking at, knowing timber rotting fungi as I do, to me it was suggestive of, there's a fungus called poria, and it used to be a big problem in mining operations here in Canada, probably in the northern United States as well, where they would use timbers to support the mine, and of course, as they went deeper, things got moister. And with the added moisture, they would get rot on their timbers, and poria was well known to be a significant problem in mining operations. So I didn't get a chance to look at it. I'm only saying it's suggestive of poria, but that morphology that we're looking at, that's very much a poria type morphology. I can't confirm that though, but just to look at it, I mean, isn't that amazing? It's beautiful and treacherous at the same time, right? I mean, it's you don't want your building to be eaten by a fungus, but I don't know. There's a beauty to it as well. All right, but with the beauty, there's also tragedy, and especially with rot. When we think about the impact of mold on the indoor environment, most people are worried about respiratory issues, the aggravation of asthma. But in Berkeley, California, this is in the United States of America, in Berkeley, California, in 2015, 6 Irish students and 1 American student died after a balcony collapsed. And the chief suspect in the collapse of that balcony was reported to be dry rot. And it was indicated, and I remember reading the articles at the time, and there's a Wikipedia page on this. You can check it out for yourself if you're interested. But there was mushrooms said to be observed growing on the balcony. Okay. So, a lot of these things are interesting as when they're theoretical, but, sometimes the reality of these things can come and impact us. I didn't get a chance to look at what happened in this particular scenario. Again, you can see the Wikipedia page for it, but it is alleged that it was dry rot that caused that balcony to collapse, and 7 young people lost their lives. And that's right in the United States of America. That's in Berkeley, California. So, it's important always, and I always maintain this with the people that I work with, clients and colleagues, to never be cavalier and always be professional, always appreciate that there can be, if these issues are not managed correctly or effectively, there is the potential for tragic and lethal outcomes. And that's a sad thing. That's a real loss of life. Okay, so that was basidiomycetes. I want to move now to ascomycetes. Ascomycetes impact the environment a little bit differently. The affected materials, again, are usually with cellulose content, so wood and paper. The decay tends to be limited to the surface of building materials, so we don't get a lot of very deep mycelial structuring or rooting typically in the built environment with ascomycetes that grow. And a lot of these species will grow with only a very minor water intrusion. So basidiomycetes tend to have a much larger requirement when we think of those rots. They tend to require a much larger water source. Ascomycetes often are willing to grow and able to grow with a smaller availability of water. And the fruiting bodies, of course, tend to be smaller. That's not surprising. If there's less food and less water, your fruiting body will probably be more modest or smaller in size. So I've got a couple of pictures of here, microscopic images. Chaetomium. Chaetomium is a common indoor ascomycete in its sexual state. So again, we're going to distinguish here between asexual and sexual growth. It's a parathecial ascomycete. I've got a picture there of hypocrea. Hypocrea is interesting, and I don't know if a lot of you encounter hypocrea in the field. It often parasitizes basidiomycetes. So often when we're collecting in the field on the weekend, polypores, I often find it on polypores. We often find hypocrea growing on polypores, and it's got really cool ascospores because it produces a type of ascospore called a part ascospore. So initially you might have 8 what appear to be 8 spores, but then they subdivide and become 16. And you can kind of see when you look at the image of those structures, of those spores, those part spores, that they are associated with each other. And you can sort of visualize how each one divided into the two. There's Eurotium there. Eurotium, we now generally call a lot of these fungi by their sexual state names because they're more common. So, you know, Aspergillus, but Eurotium would be the teleomorph, the sexual state of a particular species of Aspergillus that we find growing indoors. It's a cleistothecial ascomycete. So you notice in the structure there, it just tears open. For those of you that are interested in ascomycetes, it just tears open. It's a little bit different than Chaetomium, a parathecial ascomycete, which actually has a specific opening, a pore in its fruiting body to allow spores to escape through. All right, so our 2 common indoor fungi, ascomycetes, that fruit are Chaetomium and Pisiza. And a lot of you would know Pisiza because we do find it in the field on the weekends. In the spring right now, when you go out for your spring collections, Pisiza, various species of Pisiza do occur in the field. All right, this is a close-up shot of Chaetomium. You can see it almost has, you know, it's a very almost pubic sort of appearance. The structures, the hairs that grow from the parathecium, that long, almost elephant trunk-like structure you see extending, you know, it produces its spores in the same way. If you squeezed a tube of toothpaste and the toothpaste all came out, that's very much how the parathecia of Chaetomium produce their spores. So there's an opening in the parathecium, or you know, a round opening, and then the spores all come through that point, and they come out, and they very much stay together in that structure that you see there. And there's a number of reasons for it. I think it's mostly related to the insect vector dispersal, but that's a talk for another day. Okay, there's here on it. This is on furniture. This is on someone's chair. Oh, this was on a dining room chair. So I went to a cottage, and there was the cottage. These people had this great idea. Instead of turning off the water, they left the heater on all winter. That was their strategy for the cottage. Great strategy, except the power went out. So the power went out and all the water lines froze. And it was fine while the water lines were frozen, but then they defrosted. And of course, water flooded the environment. So the whole place was quite a bit of a write-off. So this is the ascomycete Pisiza domiciliana, named because we find it so commonly growing in human-built environments. And again, this is, look at, if you see it there, that's growing on someone's dining room chair. It grows on grout. It grows out of floors. For the Mycological Society of Toronto, for the cane foray, annual cane foray, we've actually found it growing in the bathroom of one of the cottages. You can actually see this fungus recorded for our cane foray because it was growing in the bathroom of one of the cottages, one of the participants in our annual cane foray. It grows in cars. You know, people have found this growing out of the carpet on the floor of their car. Usually it's a sign that there's a water incursion through the base of the car, so it's getting, the carpet itself is getting wet. You can find it in so many places. Domiciliana, though, Pisiza domiciliana, it's very much associated with human structures, human built environment. I never saw this ascomycete, except, thankfully, and I'm so grateful, this came in on a drywall sample. So the structures we're seeing here, and it's 1 millimeter. Boy, I'm trying to think for the American side of things. a millimeter. That's got to be, you know, was that a 16th of an inch, something like that. Anyways, it's a very small size, 1 millimeter. And it's something you're really only going to appreciate under the microscope. And I never saw it in the field. I've never collected this in the field. And I've collected an awful lot of Ascomycetes in the field. I've never collected this in the field. I only saw this from drywall. So I wouldn't know this fungus personally had I not seen it growing on somebody's drywall. I'm sure they were less grateful for it than I was. So Patilaria atrata. This is a picture here under the microscope of the ascus. So the sac containing the spores. It's an ascomycete, and the ascospores. So those are the spores within the sac. There'd probably be 8 spores in there. Typically, the occurrence of ascospores within asci, you know, are a multiple of 2. So you're going to get, you know, 4 or 8, 16, 32, some multiple of 2. All right, I want to look quickly at insect damaged wood, because this is an interesting one in the built environment as well. There's a lot of fungi associated with insect damaged wood, but they're primarily, from my experience, ascomycetes. And the insect damage is often occurring in the trees prior to them getting at the sawmill. This is an interesting thing, and we can deduce this. We can, again, put on our Sherlock Holmes hats, and we can deduce this by looking at the impacted timbers. And we're going to look at that in a second. The insect damage may happen after installation. You know, termites, carpenter ants. There's an awful lot of insects that do impact our built environment after it's been built. We look generally for boreholes, beetle labyrinth, dark, patchy discolorations in the wood. These are often our best indicators. And insect damaged wood often has fungal growth sooner than adjacent undamaged wood. And this is really an important thing that comes up, because oftentimes when there's a short-term duration moisture incursion in a built environment, so we've got a flood, we've got a high humidity situation, you know, a burst pipe, we don't often look at healthy timbers if it's a very short duration impact event. But if the timbers have been impacted themselves by insect damage, that can accelerate the timetable for the growth of fungi. So we have to look at those a little bit differently. All right, so under the microscope, from my experience, again, the species of fungi we commonly find tend to be from the genera of Ophiostoma and Graphium. When they show up on air samples, you know, when people, it's very common for people to get air samples in their homes, in their offices, wherever. These spores, though, don't tend to show up very specifically as those things. They will show up maybe as ascospores, as Aspergillus or Penicillium, or an unknown fungal spore. And we need to actually look at the affected material to get a better feel for what's growing on, because we want to see not just the spores, but also the fruiting bodies. You know, knowing how the spore is produced is much more important often, so much so than just seeing the spore by itself. So we see a lot of these ascomycetes in wood framing. I looked at a residence with a flooded basement. Mold damaged drywall and flooring had been removed. So we knew that the area was impacted by water, and moldy drywall was all removed. Moldy flooring, you know, carpeting, wood flooring, whatever it was all removed. So it was stripped back just to the framing. This is very common. We strip it back to the framing, you know, but they kept collecting air samples, and we kept getting all these ascospores and air samples. So I was called into this environment. Is that, well, we can't figure out how to get it to work. Can you come in and have a look at it? And we were getting these high counts, over 1,000 counts per cubic meter of Graphium-like spores. So what's the problem? All right, so this one's a little bit out of focus, but here is a 2x4, you know, a wood stud, you know, so the wall, the framing and most of our walls around us, if it's wood, it's a 2x4 stud. And you can see there in that stud that we've got insect labyrinth structures. Here's another, this is interesting. I always love this picture, because you can see in this picture that the board has been stamped, you know, somebody's, you know, it's actually been stamped. Oh, it's good to go. Or, but you know, simultaneously, my goodness, I can see all the ants or beetles, whatever the particular insect was here, they're actually lying dead in it. And you can see that by those structures, you know, the wood has been impacted by these insects, you know, while it's been standing, and it's been milled. So it's actually been cut post insect damage. So in a mill, there's supposed to be an individual, a quality control individual, who would pull timber like this and say, yeah, we're not going to cut this and send it out because it fails our quality control check. But this one clearly did not get pulled for quality. It got milled, it got cut, and it was sent out. And even the carpenters, carpenters building your home, putting in these timber structures, they're supposed to do their own quality checks and remove these boards. Carpenter didn't do his quality check either, or her quality check. So this board actually got installed, in a home, but it really shouldn't be. That's a fail. That's a quality fail for a timber structure. But it beat the timber mills quality program, and it beat the home builders quality program. So when we see often fungal growth on wood framing, and I see this, by the way, I go to a Home Depot or Lowe's in Canada. Here we have a place, Home Hardware. I go to these home hardware shops, these various hardware shops. I often see timbers like this available for sale. They're right there. You can go grab 20 of them and throw them in your cart, but they're already impacted by insects. Typically, this type of growth here we're seeing is typically associated with insects that are working within the wood. We don't see it on the surface of the wood, but we're typically seeing it within the wood. And what's confusing or confounding about it for a lot of the investigators in these scenarios is that, it's a board here, it's a board there. It's not all the boards, it's just a couple of boards, but you really have to stop and examine all of the boards to see the affected ones. And then so when we do a close-up microscopic evaluation of the surface of the wood here, you can see those parathecial structures. So what you're seeing there is the fruiting bodies of ascomycetes, and a lot of them are rampant. So a lot of them are just below the surface of the wood, or just buried in the surface of the wood, and they grow out of it. And those tubes that you see growing out are spores, just long chains of spores being exuded, being ejected from those parathecia into the environment. And a lot of them, of course, become aerosolized, and that's how we know that they're there. In this particular case, we thought it was an ophiostoma. Okay, so molds. So I'm only going to spend a very brief time on molds because I've already, I think, overstayed my welcome a little bit. We're closing in on an hour. Molds. So molds are anamorphic fungi. All right. So again, this is asexual growth, and molds grow on a wide array of substrates. It really does. We probably find molds growing on the widest range of home-built indoor environment materials of all things. There's so many species that will grow. They often don't need a lot of water. You find these things growing in your bathroom, on the ceiling, on the grout and the tiles in your bathroom. They're growing in your sink. If you look down in your sink for your bath, in your kitchen, in your bathroom, you often see these things growing in your sink. And what we're seeing, those asexual anamorphic fungi are typically ascomycetes. So there's very rarely do we get asexual basidiomycetes growing in those environments. It can happen, but most commonly they're asexual ascomycetes. And the great value and benefit of asexual reproduction is you can produce a large number of spores, you can produce them fast, and you can produce them cheap. It's energetically inexpensive. So without a lot of water or ongoing water, you can often get an awful lot of spore production out there. These are just sort of standard sort of stock photos of fungal growth, mold growth. So here we're seeing just masses of colonies. You can sort of visualize there. You can visualize the colony growth on the affected material here, which is drywall and a shelving unit adjacent to it. There's a very bad damaged area here, so we can see water's actually come through the ceiling. The insulation, that's fiberglass insulation's impacted. The ceiling is impacted. The walls are impacted. And that's all mold. By and large, there, when we look at that under the microscope, everything you're looking at there is mold. It's fast, it's cheap, it's energetically inexpensive, and it's the most common thing we do find growing indoors. Zygomycetes. I love zygomycetes. I think we always think there's a zygomycete we encounter in the woods. I think they call it troll's beard, sometimes growing on mushrooms. Zygomycetes aren't proper fungi in the way that we think of ascomycetes or basidiomycetes, but they share, I think, the same ecosystem and a lot of overlapping interests. We certainly see them in our kitchens, growing on bread, growing on jam. They produce structures called sporangia that are filled with asexually produced spores. They're called zygomycetes. They produce a sexually produced spore, a zygospore, but it's very rare that you would see that. I see it very rarely in an agar plate, but it's a very rare and uncommon thing to see. And the characteristics of the sporangia and the spores do help to identify the organism. Here's a group of zygomycetes, or a couple of genera of zygomycetes here. We've got Mordiarella, In the centerpiece there, they're showing the zygospores of a zygomyces called Obsidia. And on the right, you've probably bumped into rhizopus, whether you know it or not. On the right, that's a more common kitchen zygomyces that shows up growing on foodstuffs. But again, it's not a proper fungus, but those of us who look at fungi often encounter it. So I'll take you quickly through a confounding case of rhizopus. I worked on a job. It was a building, and it was an individual who had many animal companions. Now, when I say many, I want to say there was a number of rabbits and dogs and cats. It was one of these homes with just an awful lot of animal companions. And the homeowner did not seem to mind there was an awful lot of fecal matter, they just deposited around the home. I don't know, like I wouldn't want to live in a place like that, but the homeowner was fine with it. But they had a water incursion. And then so the impact of that was, a lot of this animal feces, grew, had fungal growth associated with it. Not an ideal thing. So anyways, the remediation contractors came in and they cleaned it up. So all the animal companion, the planned animal presence in the house, they cleaned up the fungal growth associated. Well, it's a zygomyces growth associated with that. But afterwards, they still had all this rhizopus showing up in the air sample. So they called me and said, we don't understand. We cleaned up all the animal feces. We still got all the rhizopus. But it turned out, and in poking around the place, there was a drop ceiling. And then so I got up and I removed one of the ceiling tiles, and they must have had just families and families of mice working up there. And the drop ceiling tiles were just absolutely covered with mice feces. So unplanned animal companions maybe. And that mouse feces, because of the elevated humidity that occurred in the space after the water incursion, it was all covered with rhizopus too. So they had to take down all the ceiling tiles, and they don't even bother cleaning them off. They just replace. It's much cheaper to replace those drop ceiling tiles than it is to clean them. So they just all went in dumpsters. But that's where the secret reservoir of rhizopus was hiding, was up in the ceiling. Myxomycetes, I'll touch on very briefly. We do find I love slime molds. I have a very fond spot. I do a talk on slime molds if anyone's ever that interested in them. We bump in, if you look at mushrooms, you're going to bump into slime molds. There's a wide array of them that grow indoors. Hockey rinks. And so this is going to appeal to those of you in the Northeast. You guys have a lot of hockey rinks down in the Northeastern United States of America. In Canada, we've got plenty of them too. Stemonitis. There's a myxomycete called stemonitis. Commonly, I don't know why, but it just seems to really do well in hockey rinks. I find it there. I've found species of Physarum, Stemonitis, Trichia, Hemitrichia, and Fuligo from the indoor environment. So there's a wider range of myxomycetes that grow indoors as well. There was a species of Physarum I only ever found in Canada in a winery. It was growing on a biofilm in the winery. There's a guy in the States at University of Arkansas, Steve Stevenson, I used to work with, and I remember sending it to him. And it's the only time I ever found it in Canada. I wouldn't have found it if it wasn't for a winery having a problem with a biofilm. So, it's interesting in a lot of these environments. It's your only chance sometimes to find these organisms growing. All right, I'm going to wrap it up there. I think I'm at an hour, so we'll kill it there, and I'll take questions or comments.
Michael, I'm absolutely fascinated and just a little bit horrified. Well, that was great. That was just some amazing examples of fungi growing in unlikely places, or maybe likely places. So we have a whole bunch of questions already, and I encourage you all to add more questions to the chat. First of all, I'm going to make it so that people can unmute themselves. So Andrew, yours was the first question. Andrew Dalsimor.
I was just curious, in the church pictures of the brown rot on the wood, how long a process is that for that to occur?
That's a good question. I've never really seen it timed. Maybe somebody's done research on that to see, from start to finish. How long does it take for any particular brown rot under ideal circumstances to consume all the cellulose or almost all the cellulose out of a timber? It's probably related to the diameter of the timber, the availability of moisture. I bet there's a number of variables that would be connected to that. I don't think I've ever seen, though, that type of significant degradation that hasn't been at least a minimum I want to say of 2 to 3 months in the indoor environment that I've observed. It's typically when we back trace, when did the first initial water incursion occur? It's typically 2 to 3 months. It's not a very fast occurrence. It tends to be they're in for the long haul. To get that type of digestion, it tends to be for the long haul. But there may be research out there that indicates otherwise. So I would look into that first.
Great, thank you.
Okay, Maricel, you didn't have a question, but you had a comment.
Can you hear me?
Yes.
Okay, I just wanted to say that Serpula lacrimans is not a polypore because it doesn't have pores on the surface. It's considered a crust fungus.
Okay.
Yeah, it has like a merullioid surface.
That's right, Marioid. Yeah, I think that comes from that. What was it? Was it the Minotaur? Was in the maze? So is that maze-like surface properly? Yeah.
You said that the fruiting body was really big, like 3. Oh, I can't remember, like orangey, brownish.
Yeah, the one I observed was about 3 feet in diameter.
Oh, 3 feet, yes. After you talk about Armillaria melea, you talk about Serpula lacrimans.
Oh, the 3 feet in diameter is definitely Serpula lacrimans. Yeah. Armillaria, I've never seen an Armillaria species that big. Well, Armillaria gets pretty big as a single organism.
No.
Yeah,
No.
But an individual fruiting body, I don't think it's to be that big still.
No. I meant is to remind you that you were talking about Serpula lacrimans after you talk about Armillaria melea. But you said that it was
they're both basidiomycetes. So what they share in common is that they're basidiomycetes.
Yeah, they do. Yeah.
Okay. Okay, great. So David, you had a question, and Tristan, I think you had a similar question. So I'm going to let David ask it.
Well, yeah, my video and sound is going in and out, but I have a big problem with dry rot in my basement from a pipe that's leaked for many years. I discovered it, but it still seems to continue to do it. And so will the fungus continue even though there's no more water?
Oh, okay. I think he cut out, but I think
will the fungus continue to eat the floorboards and the sub? Yeah, did I cut out? Sorry.
No, we heard you.
Sure. So that's part of the thinking with some of these dry rots, that once they're established, simply the digestion of cellulose, they're sometimes able to. The idea is that they're able to liberate water in the digestion of cellulose. So they're actually not just getting, the carbohydrates, the sugars from the digestion of the cellulose, but they're also simultaneously liberating water that they're able to draw in. And then as we saw in the church, if it's sending rhizomorphs out into the environment, it can actually penetrate through adjacent structures and draw water in from outside the immediate structure on which it's observed.
Yeah, I have, there's black rhizomorphs there, very prominent, but I guess the solution really is going to be to rip it all out and replace it with stuff.
Yeah, it tends to be when you look at the remediation strategies for rot, it's like brown rots and dry rots. It tends to be, find go as far as you can find, an affected area, and then cut like another 1 or 3 feet beyond that, appreciating that maybe you can't observe some of the affected area beyond what you can observe. So it tends to be a very stringent and aggressive approach to remediating it.
Well, I think that's what's needed here, unfortunately. But, we'll see. It's buying a foreclosure, you discover different things,
Absolutely, yeah.
All right, the next question comes from Ethan, and he's wondering if you could share...
Great speak, great talk.
Thank you.
So Ethan wonders if you can show off a basidiomycete that he grew, that was growing in his basement. Would you be open to that? It would require you to stop sharing.
Okay, so sorry, I'm just trying to see here.
There should be like a stop sharing. Yeah. So
Ethan,
Ethan, are you ready to share your basidiomycete?
Yes. First of all, great talk. Thank you, Michael. I really enjoyed it. And I just want to mention that we see Patullaria atrata all over New York City. It's very common in the environment. It's almost hard not to find it. So I wonder if there's sort of a distribution range that tapers off as you go north. This is going to be one of those questions that's kind of like, have you ever seen that? Isn't that cool? And also, but this is not my basement, but it's a friend's basement. And I see, just from these pictures, what I interpret as something that could be maybe a lepiotoid mushroom, a basidiomycete, growing. It's a little hard to get a sense of where it's coming from, but it seems like what you're seeing on the bottom half of the picture is the top of the foundation wall covered in sort of mortar dust and debris. But then maybe there's a beam above that could also be involved in the growth of this mushroom. And it's so, you know, I'm wondering if you've ever seen anything like this, if this is something that's common, if this is something that's unusual, if we should be sequencing this to see what it is. And if there's anything interesting other than that to say about it.
Yeah, I have to say, you know, just from looking at it from the morphology of it there, I have a hard time putting a name on it. It's very, you know, I have a terrible, I think it's attractive. You know,
Yes.
I know people hate the, I grew in my house is a ugly, terrible, horrible thing. You know, it's very attractive. You know, and I'm trying to, in my mind, they're just looking at the image, trying to reconcile. Obviously, the fungus is white, but is the area around it that's white, is that painted white, I guess, or is it is it efflorescence? You know, sometimes we get in those spaces, you know, efflorescence or something coming out of stone or concrete, or I don't know. It's tough. It's tough to say from a picture. I'm always very cautious with pictures. You know, there's only so much you can do with just a picture alone. Oh man, if I had one of those gills and some spores, I could do so much more, right? So.
Right. Okay. Thank you again.
I don't see a lot of that. I'll tell you that for nothing. I don't see a lot of that's fairly unique, but so.
Well, I'm hoping it'll come back around and I can get a sample next time.
Yeah.
Again, thanks
cool.
for the talk. It was very fun.
Thank you.
All right, more questions. So this one came from Rebecca, but she's not actually in the audience anymore, so I'm going to ask it for her. Are various species either sexual or asexual, or do various species reproduce differently in different contexts, like plants?
Yeah, so, you know, so a very good example. So in the indoor environment, a very good example of this would be Scopulariopsis, which is the asexual state of Microascus, which is the sexual state of an ascomycete. And then so typically, again, so this is an organism which, when the resources, I would say, are plentiful, you know, if it has access to a good, you know, wood substrate and maybe longer term moisture, I'll see the sexual structure of Microascus form, you know, in the surface of wood. But it's also opportunistically able in less opportune environments, maybe in carpet or in cloth or materials, to produce the asexual state, which is Scopulariopsis. So it does seem to be the fungus is able to determine, based on the availability of resources, should I put in the energy and the effort to produce my sexual structures, which, you know, more costly, or, you know, maybe I don't have a lot of resources available, I'm just gonna, you know, go asexual, and it's fast and it's cheap and it's easier to do it like that. So it does seem to be, and it does also help too for an investigator, guide where to look in the environment for the affected materials.
Great. So the next question, or actually two questions, I think, from Robbie. Robbie, are you ready to ask your question? Are you unmuted? Okay, I'm going to ask them for you. So your first question was, what happens to people living in these environments?
So with mold environments, you know, it's very individual. You know, there's people who live around mold who don't seem to be much affected by it. And there's people who live around mold who seem to be greatly affected by it. So it seems to be, the response seems to be very much driven often by the individuals in that space. There's certainly some spaces I wouldn't want anybody to occupy. And certainly insofar, and that's just thinking about respirable fungal issues, you know, mycotoxins or opportunistic pathogens. But in terms of structural deficiencies, of course, I mean, it doesn't matter who you are. You wouldn't want to be standing on a floor that's going to collapse because all the joists have been eaten by a brown rot fungus. So, there's some things that impact all of us, regardless of our age or our health or how immunocompromised or not we are. So, yeah, I mean, they're all worth addressing. I mean, I think, generally the approach to fungi when it comes to remediation and remediation guidelines and protocols, it's not that there really is no acceptable amount. Generally, if we find indoor fungal growth, we want to eliminate it. That's a general practice and approach to it.
Right. Okay. So the follow-up question was, and I think I know the answer. Can a house left uninhabited for 25 years ever be habitable again? Joists and beams have dry rot, 1860s plaster covered with mold, raccoons inhabited house, and mushrooms were growing on feces. Can this ever be a healthy house?
Well, it's interesting, and I'm going to tell you, there's historic buildings. We've had this here in Canada. I'm sure it happens in the US as well. There's historic buildings that are impacted, and it's challenging, because I've seen some of these historic buildings stripped right back to nothing but a brick wall, just brick. And then and then all the timbers that have been removed that were centuries old timbers that were decimated by fungal growth, all the plaster and lath is all gone, and there's just one brick wall. But the historians want to save that brick wall, and the thing is you build a new space in, and you walk into it, and it still smells like mold, and the challenge often is that brick or mortar or stone often holds these chemical compounds, microbial VOC, so volatile organic. Before variants of concerns took over the term VOC, I'm still mad at COVID for doing that. I always know VOCs as volatile organic compounds. So a lot of these chemical compounds, when a structure like a brick wall is adjacent to fungal growth for a long period of time, it takes up a lot of these volatile organic compounds, and then you remove all that fungal growth and you build in fresh space. But guess what? Now the brick is emitting those volatile organic compounds. So it still smells moldy. You got rid of the mold, and there's no mold growing on the brick, but it still smells like it. And you get with books as well. If you get a book, I get a lot of old books, from antique bookshops or whatever, and as a book's 100-year-old book, 200-year-old book, and the book is fine, but it's probably been stored in a space adjacent to a moldy book. So every time you open up the book, it's 150 years old, it smells like mold. But I can put it under the microscope, I'm not going to find any fungal spores or hyphal, but the smell, and that's fungal microbial VOCs, they're in the book, and the book is now a reservoir for them, and the book is emitting them. And of course, we're going to smell that. And some people indicate that seems to impact their health. Maybe they get red eyes or runny nose from that. So microbial VOCs could also be potentially a human health issue as well.
Wow, that explains a lot. All right, Kevin, you had a question. Do you want to ask it? Kevin, do you mind unmuting yourself and asking your question? Oh, has no microphone on the computer. I'll ask it for you. I have no evidence of any problems, but a burning curiosity. How much of my house's microbiome can I explore with DIY techniques?
So how much of his house is
a microbiome? If I were to play detective in my own home, how much of it could I explore using DIY techniques without bringing in the experts?
Yeah, it's well, there's the areas in our homes that are commonly impacted. So areas of high moisture. So that tends to be, of course, anywhere where there's plumbing. So kitchens and bathrooms. If you have a basement, anywhere, if you've got a crawl space, your attic, any of these places you can go where we think about the potential for failure of our built environment. Roofs leak, they do. They have windows leak, they're put in brand new and they're very well sealed, but 25 years later, they're not as well sealed as they were initially, and you have some water incursion in and around them. Bathrooms, plumbing, it just happens. If I can't imagine that there's a home in North America that if I went to it, that with some good, solid detective work, I couldn't ultimately find some fungal growth in it. Now, some of it may not be active, mind you. Some of it may be, like when we looked at those timbers, some of it may be fungal growth that existed prior to being built in, and there's no current moisture source. So it's historic fungal growth, but it would be fungal growth nonetheless. And then over time, I mean, it's almost inevitable. I think all buildings will experience some amount of fungal growth. The question really boils down to, is it being reasonably mitigated, and is it impactful on the health of the occupants?
Right.
Yeah.
Okay. So, I mean, if we ever get bored, or if there's ever no fungi in the woods, we could just start at home and see how far we get. All right. So, Michael Moser, you had a question.
Yes. So, the kind of pleasant smell of a cottage, which is must, is that fungus?
Well, that's a good, it's an interesting thing. Fungi often get fingered or blamed for odors. Bacteria are much smellier than fungi. And I'll tell you that for nothing. I know I have worked with bacteria. I don't like bacteria at all. They are very, there's a lot of very smelly bacteria, and there's a lot of overlap. So just because there's a strong smell, that musty smell, that skanky smell, in a cottage or in a bottle of wine or whatever it is, it doesn't necessarily indicate, fungal activity. It certainly could, but not necessarily. And then also, again, remember the VOCs issue. So it could be representative of a historic problem. So maybe once upon a time there was a fungal issue there, but they addressed it, and but now the smell simply persists. I remember, I think it was my mom. She had a water incursion and some fungal growth, some mold growth in her basement, and it was addressed. And the place always smelled like mold until she got rid of the rugs. And once she got rid of the rugs, well, there went that mold smell. The rugs were a reservoir for, I think, the microbial VOCs. So the rugs always just smelled like mold. You could look at them and they're not technically moldy, but they seem to have absorbed and then re-emitted the VOC smell, the smell of mold. And once they were gone, man, that place was entirely, scent free. So maybe more tile floors in place of carpets and rugs would help a lot of basements and cottages. But
What about linoleum?
Well, linoleum is probably fine, but a lot of times you can get microbial growth beneath it. So, so a lot of times we find problems that are either beneath the subfloor and the flooring material, or then just flat out underneath the subfloor. So, sometimes you got to kind of go under and have a look, see what's going on.
Okay, this was such you were a delight to listen to. It's always a pleasure to hear somebody who loves what they do, and it must be a kind of happiness to love something that you are always you're always going to find something to love, right? Because
I thank you. It is rewarding. It's enjoyable. I get to be Sherlock Holmes. I guess that's my favorite part of it, is putting on the Sherlock Holmes hat and being the investigator and solving what is the problem, how do we get to the roots of it, and then finding it, how do we solve it? It's enjoyable. I don't know. I find it enjoyable, so it's rewarding for me.
That was clear. Thank you.
Thanks.
All right. And now we had a question from Roman, and I'm sorry I accidentally jumped you. So Roman, do you want to ask your question now?
Yeah. Hey, Michael. You partially answered this, I guess, about remediation, but we just had a situation in our home where we had a bathroom leak come down in our kitchen ceiling. So all kinds of damaged drywall and wood and OSB subfloor. So we cut out everything that was visibly damaged and moldy. Anything that was sort of visible on the floor joists, I couldn't take out. I sprayed the heck out of with, I think, consortium or some mold killer. How effective is that? Like you say, you probably can't go far enough, but how far is good enough?
Yeah, I mean, the key thing always is cutting off the moisture. Once the moisture problem is addressed, most of the other issues begin to abate on their own, because you don't have active growth anymore. So step one is address the moisture issue. So if you've addressed that, that's the biggest and most important step. And then in terms of removing impacted and affected materials, insofar as you've done that to the best of your ability, that's the correct thing to do. So one of the problems with the idea of applying a fungicide, so a chemical agent that will kill fungus but not remove it. So the thinking is this. So if the fungus, if what's present of the fungus represents a chemical hazard, so in other words, if there's toxic components, let's say a chemical toxic component within the fungus itself, if you kill the fungus, well, that chemical threat would still exist. That chemical hazard still exists. The only way you'll eliminate a toxin issue associated with the fungus is by completely removing it. Killing it is insufficient to deal with the problem. If it's a pathogen issue, then yeah, killing it deals with the problem. But if it's a toxin issue, the killing it isn't really, sufficient. You really do have to remove it in order to effectively address the potential toxin issues, if that makes sense.
Yeah, that's great. Thank you.
All right, I had a question as well. I'm just fascinated by your job. You get a call and then you show up, and I'm so curious about what you do. You talk about, counting spore counts and, looking for spores. I mean, what sort of some of your equipment that you use to sort of figure out what's going on in a particular site?
Oh yeah, I have an awful lot of microscopes.
Mm-hmm.
And I mean, generally speaking, it's when I'm called, I mean, a lot of my work, I work with a lot of most of the work that happens is fairly standard. So generally, I'm often only employed on challenging cases. If it's simple cases, nobody calls me. I get called in when somebody's having a problem with a persistent issue and they can't solve the problem, and then so they would maybe bring me in. And generally, I often begin to do diagnosis without even visiting the site. Somebody sends me air samples, and I'll look at them microscopically with a microscope and evaluate them and look at the qualitative and quantitative aspects of fungi on them. Sometimes there's other particles present as well. Sometimes there's other issues. I might look at a sample and say to the person that sent me, oh, I've got a client and they've got terrible allergy issues and we think it's mold. They send me the sample. There's really no mold on it. But my goodness, I can't hardly see the sample for all the animal dander. There's an awful lot of animal dander on there. What's going on in that environment? And oh yeah, the guy who lives there has 7 dogs. And it's carpeted from corner to corner. Well, I don't think it's a mold issue. I think if you've got 7 dogs, you might want to give up corner to corner carpeting or vacuum a lot more often. That you've got maybe potential other allergen issues operating in that space. But and generally they only call me out to site, especially now with COVID. I guess COVID's been terrible. What a terrible thing for my business model. I guess nobody wants you on site anymore. They are, you stay home, don't come here, we don't care, So it's challenging within the COVID type of environment. There's a lot less interest in having on-site analysis done, but I still get called out on the challenging cases. But it's like, it's as I say, I get called in for the tough jobs, not the easy ones. They don't need me for the easy ones.
Right.
They call me on the tough ones, right? So.
That sounds like an amazing job. I don't think you're ever going to be bored. Thank you so much for an absolutely fascinating talk. I think we're all sitting here somewhat awestruck because this is just a way of looking at fungi that I don't think we've ever had before. So it's really eye-opening to realize that there's fungi all around us. So yeah, thanks for an amazing talk. Really appreciate it. Lots of people left comments in the chat saying this was mind-blowing. So thank you.
Well, it's my pleasure, and thanks for having me out. Susan had been trying to get me out for years, and our schedules never seemed to line up. But this works a lot easier because instead of me flying down to Boston or something like that, I just get to log in from Toronto. So that's an awful lot easier. But I'll tell you something. I miss the in-person stuff.
Agree.
And and especially doing forays. And there's a gentleman, Ethan, there was saying they get all sorts of patallaria in New York. Now I'm jealous. I got to come to New York.
You should.
I only got to see it once. That's not fair. Maybe global warming will bring it up here. If there's some good from global warming, maybe we'll see more patallaria in southern Ontario. But in the meantime, I'm just waiting, right? So you have to sometimes get out and get around to see some of the other interesting fungi out there, right? So.
Totally agree. Well, you have a talk on video games and slime molds. That sounds very tempting. So yeah, we'll consider you for next year. But thank you today for just a great talk.
Well, thanks so much, and have a great night. Thanks everyone for coming out.
Yeah, thank
Thanks
you.
very much.
Okay. All right. Bye.
Bye-bye.
Bye, everybody.